Hello everybody. Today's topic is how can you make ATP in the absence of a sufficient amount of oxygen? If you remember yesterday's discussion, oxygen is necessary as the final electron acceptor at the end of the electron transport system on the inner membrane of the mitochondria. And if you don't have oxygen gas available as a final electron acceptor, the electron transport system will stop running. If the electron transport system stops running, hydrogen ions will stop being pumped from the matrix into the intermembrane space or outer chamber. If that happens, we lose our gradient across the inner membrane, and there is no motivation for the hydrogen ions to diffuse back into the matrix through ATP synthase. As a result, we get a much diminished ability to make ATP. We call ATP production in the absence of oxygen anaerobic. And this study guide I'm showing you here is actually from my advanced biology class, but we're going to use it because it'll work. And we're only going to concentrate on how humans would do this, not bacteria and other things. So we're going to concentrate on the lactate fermentation, or on the right hand side. But it is true, if we look here, that anaerobic pathways do not yield as much ATP per glucose molecule as you would get via aerobic processes. Matter of fact, your book says anaerobic processes will only make about 5% as much ATP as aerobic processes, but 2.5 times faster. So that's what the book says as far as statistics on this. You make about 5% as much as you would using aerobic processes, but you make it 2.5 times faster. You still don't come out, as good, but that's the way it is. Now, if we look at this paragraph, it talks about how some organisms like bacteria and protists and whatnot, they use an anaerobic pathway called alcohol fermentation. We don't do that. If we did that every time we made ATP anaerobically, we would get drunk. Instead, we have a process called lactate fermentation, and I'm just going to try to simplify it here. It basically is glycolysis with a step added. So it happens in the cytoplasm, okay? In the cytoplasm of the cell. So let's start with glucose. We'll make it symbolically here with our little 6 circles, which is supposed to represent the 6 carbons of glucose. And we know, if we summarize what happens during glycolysis, that 6 carbon glucose breaks down into 2 3 carbon pyruvic acids. Now, to make that happen, if you remember, we need to invest 2 ATPs into this. We will get 4 ATPs out for a net gain of 2 ATPs, because 4 minus 2 is 2. That might be where they get the 5%, although it's more like 6% of the 36 we would expect. But maybe I did that math wrong. Okay, now also there was a need to bring some NAD in here and pick up some electrons. And then this NAD would normally go to the mitochondria and give those electrons up to the first proton pump in the electron transport system. But if you remember, because there's no oxygen, the mitochondria is shut down, so that can't happen. Similarly, the pyruvic acid, well, let's ignore that for now. Basically what happens here then is, if you can't get rid of that electron right there, which is symbolized by this H, if you can't get rid of that electron, all your NAD electron carriers would get filled up, and you wouldn't even be able to continue glycolysis. So you need a place to get rid of that electron. So a step is added onto glycolysis in which the two pyruvic acids are converted into two lactic acids. Now you could look up the series of steps that make that happen, but for our purposes I'm not going to do that. One way you could think of it is the NAD, that step is sort of, quote, created as a means to dump off those electrons, so you can go get more from up here and continue breaking down the glucose and continue to make those 2 ATPs. Now the good news is this lactic acid will diffuse into the bloodstream, get swept away, and probably eventually end up in the liver. And in the liver, it can actually be converted back to pyruvic acid, or even back to glucose, and then stored in the liver as glycogen, or in the muscles as glycogen. And then you could either break the glucose down when more oxygen is available, or if it only got converted back to pyruvic acid, you could now, when the mitochondria opens back up, send that pyruvic acid to the mitochondria for the Krebs cycle and electron transport. So what I'd like to do is talk a little bit about how this relates to a couple different kinds of muscle fiber. You have what's known as slow twitch muscle fibers and fast twitch muscle fibers in some ratio making up your skeletal muscle. You have some proportion of the muscle fibers in your skeletal muscle being slow twitch, and some proportion of the muscle fibers in your skeletal muscle being fast twitch. Slow twitch muscle fibers function only aerobically, so they would not be undergoing lactate fermentation to produce ATP. And these types of muscles are the types that engage most effectively in steady, prolonged activity like distance running, or if you were a duck or a goose, migration, long migration flights. Speaking of ducks and geese and chickens and whatnot, this would typically be known as dark meat. And that's because it contains a protein called myoglobin, which gives it its dark pigment, and that myoglobin is the protein that has oxygen carrying capacities and oxygen holding capacities. Now, the fast twitch muscle fibers will be of a lighter color, and so if we might call that the light meat. For example, if you were eating a Thanksgiving turkey, turkeys don't spend a lot of time long distance flying. So they need short bursts of quick energy in their pectoral muscles, and so their breasts have light meat. And you would find in their drumsticks dark meat, because they do a lot of running and walking and whatnot. And that would be more aerobic activity. Now, in a duck or a goose, they're not going to have light meat on their pectoral muscles because they are long distance flyers. They're going to need to have a lot of slow twitch muscle fibers there for prolonged long distance activity. And that about does it. We're not going to talk about the rest of this page right now because it's not really pertinent to our chapter.